Dear students, after a short break, I have come to you again. Today's topic is chromosomes. You have already studied the cell in class 9. You also know that living organism's body is made up of cell or cells. After the establishment of the cell theory proposed by Schwann and Sladen, a new horizon began in the study of the cytology. This theory states cell originates from pre-existing cell, and also the continuation of life occurs by cell division, and which in turn is controlled by cell cycle. Now, before entering into the cell division and cell cycle, I discuss briefly the chromosome. So what is the chromosome? Chromosome is the gene-bearing, self-replicating, rod-shaped or filamentous nuclear component, which is structurally nucleic acid and protein, functionally hereditary bearer, which transmits from mother cell to daughter cells by cell division, and from parents to offsprings by reproduction. So from this definition we see that chromosome present in the nucleus, it is made of nucleic acid, that is DNA, RNA and protein, and it has gene which carry the characters and which transmit from mother cell, that is the cell that divides is called mother cell, and the cells that are formed from the mother cells are called daughter cells. Okay, so the chromosome that present in the nucleus, how is this chromosome formed? Because you observe in a cell inside the nucleus, under microscope chromosomes are not present. You observe the interesting nucleus or interphase of a eukaryotic cell, there are fine thread-like structures present in the form of a network, which are called chromatin threads. In reality, these chromatin threads are the DNA molecules. During cell division, these DNA molecules coil round the protein molecules to form thick, short structure known as chromosomes. That is, inside the nucleus, these DNA molecules remain as long thread which present either fully or partly coil formed, and when it becomes short and thick and takes the particular shape, it is regarded as chromosome. So what is the meant by the coiling round the protein? In chromosome formation, the histone protein actively participate. These histone proteins are of 5 types, like H1, H2A, H2B, H3 and H4. Out of these, the H2A, H2B, H3 and H4, 2 molecules each form the histone octamer, and long DNA threads which coil round these histone octamer, 1 full and 3/4 turn, and the ends, that is entry and exit, are tied by H1, and as a result small bead-like structure is formed called nucleosome. So a long DNA thread which present inside the nucleus, by the formation of numerous nucleosome, it become very thick and short, and it is the chromosome. Okay, and now you see that within the chromosome, that is the specific DNA molecule, the specific position codes the particular protein, and this portion of the DNA is the gene. So now we come to the point gene. Scientist Johansen first used the term gene, and scientist Waldeyer first used the term chromosome. So what is gene? Gene is the functional segment of a DNA molecule. You know DNA is a macromolecule and is a double-stranded helically coiled structure, and the particular part of the DNA is the gene. That's a functional segment of DNA with specific polypeptide sequence present in a specific locus of a chromosome, and which expresses a character through synthesis of specific protein. Functional segment of DNA containing specific polypeptide sequence present in specific position of a chromosome, that is locus, and expresses a character through the synthesis of specific protein. It is a gene. So you observe that there is the definite relation between the gene, DNA and chromosome. So what is the relation? Gene is the functional and structural unit of heredity. Structural, functional unit of heredity. And gene is present in the DNA. A DNA contains a number of genes. So genes are the part of DNA and they are interrelated. A DNA molecule, when coils round to form thick, short, then it is regarded as chromosome. That is, chromosome is made of DNA. And as the chromosome is made of DNA and gene is present in DNA, it is the part of DNA, so they are interrelated. And you already know that the chromosome mainly formed by coiling, that is spiralization, condensation of chromosome. So they are interrelated. Now we come to the another parts of it, that is types of chromosome. Types of chromosome. In a cell, two types of chromosomes are present. Autosome. In a diploid cell, the chromosome pairs which are identical in both the sexes, that is male and female, and which gives the information the vegetative body, that is somatic body, that is structure of the body, known as the somatic cell, known as the autosomes. In human body, the first 22 pairs of chromosomes are autosome. That is, in human body, 23 pairs of chromosomes are present. The first 22 pairs are autosome, which determine the somatic body. An allosome or sex chromosome. In a diploid cell, the chromosome pair which differ in sexes and which determine the sex in individual. It is known as the allosome. In human body and also other animals, the allosomes determined by X and Y. That is, in human body, the last pair, 23rd pair, the allosome, which are determined, marked by X and Y. In male, both the allosomes, that is X and Y, are present, but in female, only X and X are present. During sex determination, we study this again, and as the males contain both the X and Y chromosome, they produce the two types of gamete, X type and Y type, and hence males are known as heterogametic sex, while female produce only one type of gamete, X type, and known as the homogametic sex. The autosomes and allosomes, that is types of chromosome. Now the chromosome number. Chromosome number. In a particular species, the chromosome number is always fixed. It is constant. It is known as species-specific chromosome number. For example, in human body, that is all human beings, the chromosome number 2n equal to 46. That is, in a pea plant, 2n equal to 40. In a Drosophila, a type of fly, fruit fly, 2n equal to 8, and so on. It is the species-specific chromosome number. And now we come the types of cell. Before types of cell, now first diploid and haploid cell. Diploid cell and haploid cell. Haploid cell. In a cell, when all the species-specific chromosomes are present in pair, that is in two sets, the cell is said to be diploid. That is the cell in which all the species-specific chromosomes are present in pairs. The cell is said to be diploid. Here, chromosome number is known as diploid number, which is denoted by 2n, and which we said before, that is in a somatic cell 2n equal to 46. That is 2n number means diploid number. In somatic cell, in human body, the chromosome number 2n equal to 46. That is, it is diploid. Haploid. The cell in which all the species-specific chromosomes are singly present, that is one set, it is known as haploid, and the chromosome number, the haploid number, and it is denoted by n, that is number of the diploid half. For example, in human, that is n equal to 23. That is, in human body, the somatic cells are diploid cell, and Human body, the gamete or reproductive cells are the haploid cell. So now we explain the somatic cell and reproductive cell. And reproductive cell or gamete. Reproductive cell or gamete. Somatic cell, the cell which take part in the formation of vegetative body, somatic body, known as the somatic cell. That is, whole body mainly made of somatic cell. Here chromosome number 2n, that is diploid, and the reproductive cell, the cells which takes part in the sexual reproduction, known as the reproductive cell, which are also known as gametes. And chromosome number of the reproductive cell is haploid. So we now come to know what are the somatic and reproductive cells present in the body. You know, in sexually reproducing organism, a zygote is formed by the union of two gametes, male and female gamete. It is the zygote, and this zygote is a somatic cell. Zygote is a somatic cell. From this zygote, by repeated mitosis cell division, an embryo and ultimately an offspring is produced. And mitosis is the cell division where a mother cell divides into two daughter cells which are identical in respect of chromosome number, that is quantity, and in respect of quality. So all the cells, that is zygote or somatic cells that are formed by mitosis are also somatic cells. So whole body of the offspring is made of somatic cells. That is, the body is the combination of number of systems: alimentary system, circulatory system, nervous system, respiratory system, similarly reproductive system. So as the whole body made up of somatic cell, so organs, the reproductive organs also made up of somatic cells. The main reproductive organ, that is testis in male and ovary in female, are made up of somatic cells. So long the offspring remain immature, the cells of the testis and ovary, that is testis or ovary, are regarded as somatic cells. But when this offspring attains the reproductive ability, that is become mature, the somatic cell of the reproductive organ, that is testis, ovary, produce a special type of cell by another type of cell division called meiosis, known as gametes. So the somatic cell that now produces the gametes are no longer regarded as somatic cells. These somatic cells are now regarded as germ mother cell. So germ mother cells are nothing but somatic cells which are predetermined to produce the gametes, and meiosis is the cell division by which the chromosome number of the mother cell reduces to half in the daughter cell. You have already studied, that is, the chromosome number of the gamete is haploid. Okay? So in this way, the living body contains the chromosomes, that is somatic cell, reproductive cell and others. And now we come to the point, the chromosome, structure of a chromosome. That is, physical chromosome structure can be discussed through physical structure, that is morphological, and also the chemical structure. At first, the physical structure. Inside the nucleus, we see the chromatin threads. But during cell division, these chromatin threads, by spiralization, condensation, gradually become thick and short, the chromosome. And under microscope, this chromosome shows the following parts. Number 1, chromatid. During interphase, last part of interphase and early prophase, the chromosome appears as a fine long threads, and which present in two thread-like structures. Such each thread-like structure is known as chromatid. They remain bind in a point. So during last part of interphase or the early prophase, the chromosomes appear as long threads and which consist of two fine thread-like structures, each thread-like structure known as the chromatid, and two chromatids remain joined or bind in a point. Now the primary constriction. Primary constriction and centromere. So along the length of the chromosome, the small region where centromere is present is the primary constriction. If you strain the chromosome, along the length of the chromosome you observe there are one or more gaps. That is, staining is not developed in these rows and so appear as gaps. And these gap-like structures regarded as constriction. And the constriction where centromere is located is known as primary constriction. So what is centromere? That is, chromatid is nothing but DNA, and centromere is the small part of DNA. Small part of DNA which is inactive. That is, part of DNA which is active is the gene. The small part of DNA which is inactive and where two chromatids bind is known as the centromere, and it is this constriction is the primary constriction. And on either side of this centromere the protein discs are present known as kinetochore. And it is the region during cell division the spindle fibers get attached. So in a chromosome, along the length of the chromosome there is a specific region which is the primary constriction. For a particular chromosome, the position of the primary constriction is always fixed, and for a single chromosome generally a single primary constriction is present. According to position of the primary constriction or centromere, the chromosomes can be divided into four. That is, if the primary constriction or centromere is present at the center, so that the two arms become equal. That is, on either side of the centromere regarded as the arm. And it is the region where two arms can fold. In no other regions the chromosome can fold. And if the centromere is present at the center of the chromosome, two arms become equal, and during anaphase movement of the cell division, these two arms hang and appears as V, and it is known as metacentric chromosome. And if the centromere is situated slightly away from the middle in any side, so that one arm is long, another arm is comparatively short, and during anaphase movement it appears as L, it is known as submetacentric. And when the centromere is situated almost towards the terminal end, so that one arm is very long and other arm is very short, and it appears as J, it is known as acrocentric. And if the centromere is situated at one end of the chromosome, so that only one arm is present and it appears as I, it is known as telocentric. So here we discuss the chromatid and primary constriction. Now secondary constriction. Secondary constriction, that is along the length of the chromosome, the constriction other than primary constriction is known as secondary constriction. That is, any constriction other than primary constriction regarded as secondary constriction. Thus, number of secondary constriction may be none, one or more, but if present, its number, its position for a particular chromosome always fixed. These are the chromatids. Primary constriction. Primary constriction. Kinetochore. Here the centromere, the chromatid, two chromatids. These are the coil structures which mainly regarded as chromomere. So it is the primary constriction, there is another constriction, it is the secondary constriction. Secondary constriction. If the secondary constriction, that is DNA, molecule contain rDNA, ribosomal DNA, which synthesize rRNA and ultimately nucleolase, then this constriction is known as nucleolar organizing region, NOR. And the position of the secondary constriction, if situated at almost towards the terminal part, then the terminal part appears like a bulb-like structure. It is known as satellite body. Satellite body. Therefore, satellite body. In the satellite body region, the nucleotide sequence that arrange repeated manner, the same sequence, and so inactive. And this satellite body, that is also when present, this chromosome is known as SAT chromosome. SAT chromosome. SAT, that is sign, acid, thymidine, or cyanoacidothionucleinico. Next, telomere. Telomere. Both ends of the chromosomes are known as telomere, where the repeated nitrogen base sequences are present. That is, these portions of the DNA molecule inactive, and that gives the definite polarity to the chromosome, and which prevents the joining of two chromosomes. It is the telomere. And now we come to the structure of the chromosome in respect of its chemical composition. Chemical structure of a chromosome. Chromosome consists of the following parts. Number 1, nucleic acid. Number 2, protein. Number 3, metallic ions. Nucleic acid, 2 types, the DNA and RNA. In DNA, the deoxyribose sugar nitrogen bases and phosphoric acid. Nitrogen bases are two types, purine and pyrimidine. Purine two types, adenine A and guanine G. Pyrimidine two types, thymine T and cytosine C. Similarly, RNA, the ribose sugar, nitrogen base and phosphoric acid. These nitrogen bases, purine, similar to DNA, that is adenine and guanine, and pyrimidine. Here, in place of thymine, uracil U present, and cytosine. And protein two types, histone and non-histone. Histone with the amino acid, mainly the major amino acids which are present in histone protein are arginine and lysine. Arginine and lysine, whereas non-histone protein mainly tryptophan and tyrosine. And metallic ions like Ca2+, Mg2+, etc. It is the chemical structure of the chromosome. So you see, that is, the chromosome consists of nucleic acid, mainly DNA, and also small part of RNA, mainly histone protein and also non-histone protein, and small traces of metallic ions. The DNA, you know the structure of the DNA, where the sugar is deoxyribose and the nitrogen bases adenine, guanine, thymine and cytosine. But in case of RNA, the nitrogen bases adenine, guanine, uracil and cytosine. It is the chemical structure of chromosome. Now we briefly discuss the structure of a DNA molecule. According to Watson and Crick, DNA is a double-stranded helically coiled structure, just like the spiral iron staircase. In a iron staircase, there are two poles, and DNA also there are two poles. These two poles are formed by sugar and phosphate, and the rungs are formed by nitrogen bases. And two poles which are present in the DNA molecule run antiparallelly. That is, if in one strand it runs from 3' to 5' upwards, in another strand the 3' to 5' downwards. That is, DNA is made of the nucleotides, deoxyribonucleotides. Each nucleotide consists of three parts: one molecule of deoxyribose sugar, one molecule of nitrogen base out of four, A, T, G or C, and one molecule of phosphoric acid. So I say that the DNA molecule is just like the ladder-like structure. The two poles of this ladder formed by sugar and phosphate. Sugar and phosphate. And as it is formed of nucleotide, and nucleotide is formed of the three structure, one molecule of sugar, one molecule of nitrogen base, and one molecule of phosphoric acid. Similarly, here A, T, G, these nucleotides are then joined together, and in this way, by sugar phosphate backbone is formed. It is the one pole, and in a sugar molecule there is the 3' and 5' end, which you know later on. Suppose it is the 3' end, that is here 3' means the 3 carbon position is free, and here the 5 carbon position is blocked by phosphoric acid. So here the opposite strand antiparallel, it is the sugar, and then here one nitrogen base, but phosphoric acid present in the 5' end, so it is the 3' end, and antiparallel. Here the nitrogen base which is placed to the opposite strand, this arrangement is always fixed. That is, adenine always binds with thymine by 2 hydrogen bonds. So here the sugar, here must be adenine, phosphoric acid. The sugar, here must be cytosine, and by 3 hydrogen bonds. There is guanine with cytosine by 3 hydrogen bonds. And now we join. So it is the 5' end. In this way, 2 strands are formed, and after the formation of 2 strands, it coils in a particular, that is mainly right hand side, at regular intervals to form such a double-stranded helically coiled structure. The distance between 2 steps is 3.4 Armstrong, and as it coils round the right hand side in a complete turn which is 360 degree, there are 10 steps. So in 10 steps, that is length of the 10 steps 34 Armstrong, and the diameter of the helix is always 20 Armstrong. It is the structure of the DNA molecule, and then RNA molecule, the structure similar. Here the DNA is the double-stranded, but RNA is the single-stranded. Here sugar is ribose, and in place of thymine, the uracil is present. So if we remove the one side, and here in place of T, U is placed. It is the structure of RNA. And in case of DNA, as the 2 strands, and adenine and thymine, guanine and cytosine are present, these are known as complementary base pairing. And here complementary base pairing is absent. The protein already I discussed, that is histone and non-histone proteins. Histone proteins 5 types, also already discussed, that is H1, H2A, H3, H4, and H2B, H3 and H4, and then protein. So this is the overall structure of the chromosome, that is physical structure and chemical structure. Now you follow, if this video is useful to you, then you share it, subscribe it, and also give the like. Okay, thank you all.